should see the screen. Yeah.
That's brilliant. Thank you. So I can see there are a few more people joining, but I think in the interest of time, we'll make a start now. So it's really brilliant to welcome Professor Marcus Kaiser today. I believe that you originally studied biology and computer science before a PhD in neuroscience. And Professor Kaiser is now a professor of neuroinformatics at Nottingham University here in the UK, where he develops new approaches to brain stimulation, really informed by computational models and neuroimaging in particular. He's obviously very much a leader in the field of network neuroscience and also neuroinformatics more broadly. And so it's really a real pleasure to have you here with us today. So without further ado, I will hand over to you. Please do put any questions that people have in the chat as we go along, and then we'll take those questions at the end.
Yeah.
So over to you, Marcus.
Yeah. Okay, thanks very much for the introduction and for the invitation to give a talk in the seminar series, just between Ed Bulmer and Olaf Spon, so it feels very nice. Um, if you know one thing about Nottingham, you know about Robin Hood. If you know two things about Nottingham, you know that MRI was invented at Nottingham University. So there was a Nobel Prize to Sir Peter Mansfield for the invention of MRI. And because of this history, Nottingham is a big center for imaging. Nottingham is hosting the National Center for High Field MRI, which means that we are getting a scanner with 11.7 Tesla for human MRI. So we can look at the human brain in much higher spatial resolution. And based on this, we recently started the Center for New Technology, Neuromodulation, and Neurotherapeutics. It's a cross-faculty center with around 30 faculty members, where we go all the way from mathematical and computational modeling to engineering and device development, to animal studies and to clinical research. So on the left of that image there, you see OPM, which is a novel way to have mobile MEG recordings. And because they're mobile, you just have this kind of helmet on and you can move your head around. So it's ideal for children and also ideal for patients who don't want to keep their head still. So how do connectomes change during brain development? We can basically observe how the brain is connected. In this case, you see the human brain based on diffusion tensor imaging, based on the special MRI protocol. We can look at the fiber track connectivity between different parts of the brain, the structural connectivity. So we can observe that there are some highly connected nodes. There were some changes in directionality. If you zoom in and you look at individual axons, there are different modules in the system. And you can wonder how those features came about. So what happened during brain development? What are the mechanisms? Are there genetic mechanisms, or is there some kind of self-organization going on that does not depend on genetic mechanisms? So there's an overview of mechanisms in this review here, a mechanism of connectome development. The features we see across species is that there are some long distance connections in brain networks and actually more long distance than would be expected. So if you would like to reduce the total energy, the total wiring in the system, you wouldn't have as many long distance connections as you actually see in the system. We also have imbalanced connectivity, where connectivity in one direction is a lot stronger than connectivity in the other direction. Or in some cases, it might be one-way street. There might be connectivity in one direction, but not in the other direction. It's something to keep in mind because the data we get from the human brain based on diffusion tensor imaging doesn't tell us about the directionality. We just assume that fiber tracks go in both directions, but this may or may not be the case. If you look at animal studies, for example, the fruit fly, we see a lot of one-way streets in the system. So there might be one-way streets in the human brain as well that we just don't know about yet. What we also find is a modular organization. A module means that you have many connections within a module and few connections between modules. And finally, we find an organization with some highly connected nodes or hubs, and also stronger connectivity between hubs, which we call the Witch Club network, stronger connectivity between hubs than would be expected. So how can we explain those features based on brain development? So first of all, where do hubs come from? So one possibility in the literature that was proposed early on is preferential attachment. So the idea is that you have some starting network and new nodes are attached to the network, but the new nodes have a preference to attach to other nodes that are already highly connected. And by this mechanism, the witch nodes, so the ones that already have many connections, get even richer, and they finally become network hubs at the adult stage. However, this mechanism is unlikely to take place in biological networks because you would need complete knowledge of the system. So you would need to know for each target region in the brain which of those target region is highly connected. How many fiber tracts does another brain region have? And it's unlikely that this kind of information can be transferred, especially over long distances. So there has to be another mechanism or multiple other mechanisms to explain hubs in the system. So one potential mechanism that can explain hub development is that during individual development, early established nodes have more time to establish connections. So we see that in this example here, if we just look at development of cortical layers, and we have different Brodmann areas here, you see that while the starting point is similar, some areas just finish developing connections earlier than others. So Brodmann area 24 is done first, and Brodmann area 17, visual cortex, has brain development going on for a very long time, so would be able to establish connections or receive connections and so on. So we come up with the old gets richer model, that early nodes are more highly connected. So if we have those nodes at the time axis, we have older nodes that are established early on, and then they finish establishing connections early on as well. They have some intermediate nodes and some young nodes that are established at the end of brain development. For the older nodes, they can connect to the intermediate ones. They can't connect to the young ones because those haven't been formed yet. But for the intermediate and young nodes, they can all connect to the older nodes. So the older nodes have a high opportunity to establish connections and to get incoming connections. And because of that, we would expect that there's, first of all, a higher degree of connectivity for older nodes in the network. And second, that older nodes relatively receive more incoming connections. For the first prediction, we can look at C. elegans. For C. elegans, we know about the birth time of individual neurons. And we can take that as a starting point to say whether it's an early node or not. So whether a neuron is established early on or later on. And we can then look at the degree, the number of connections at the adult stage. And if we look at neurons with a higher degree, so let's say above 40, all of them are established early on. They're all born early on during development within the first 500 minutes. Whereas for the later nodes, we don't see high degree nodes over there. So the first prediction is basically the case that older nodes show a higher degree of connectivity. For the second prediction, if we want to look at the number of incoming and outgoing connections, we can look at the rhesus monkey fiber tract network structural connectivity based on track tracing, based on injecting dyes that are then traveling through the fiber tracts. So we can look at three different parts of the brain. The archicortex or paleocortex, so those are structures of the brain that are established early on during brain development, but those are also structures that are older in terms of brain evolution. We can then go to later structures, parietal and occipital, and then to even later structures like frontal, temporal, and sensory motor, so regions that finish development later on. If we just look at connectivity between archicortex and parietal cortex, old and intermediate structures, we see a slight imbalance in terms of incoming connections. There are 36 incoming, but only 25 outgoing. If we look at intermediate and new areas, we see again a slight imbalance, so 83 incoming and 78 outgoing. But if we look at the newest structures and the oldest structures, we see a very big imbalance. So there were 100 incoming connections, but only 66 outgoing. So this is exactly what we would predict based on this idea that older structures get richer and have more incoming connections. But there's also a second mechanism that can lead to highly connected nodes in the network. So normally when you look at network development, all the algorithms are adding one node at a time. Okay, so we have one brain region at a time that is added to the network. So that would be linear growth. The number of nodes in the network is linearly growing over time. But this is not biologically realistic. What is more realistic is that you have cortical tissue that is then splitting up into two regions, and those two regions split up into four regions, split up into 8 regions and 16 regions, and so on. So it's more like a nuclear chain reaction where at each time step you get a doubling in the number of nodes that are added to the system or that are present in the system. So you have nonlinear growth, you have accelerated growth in terms of the number of nodes in the network. And this is something we can observe in the degree distribution. If we look at the degree, the number of connections of a node, k, and the probability that such a node occurs in the system, if we have linear growth, we get the wet distribution. So we have a peak at an early stage, and all the other degrees are relatively close to that peak. On the other hand, if we have nonlinear growth, where the number of new areas is increasing over time, we get more connections that are more brain regions that are sparser, so there are fewer connections. But on the right-hand side, we also get more brain regions that are more highly connected. So we get more network hubs by having this kind of nonlinear growth. So both mechanisms can play a role in getting highly connected nodes in brain networks without relying on any kind of preferential attachment or genetic mechanism. We can also simulate how the brain is developing at the local level. So this is a model we published 4 years ago where we look at the growth of cortical layers. We have progenitor cells, so you have different stages of cell division S1 to S7. And at each stage, a cell can either continue cell division, so go to the next stage, S3, S4, and so on, or a cell could die, so there's early apoptosis, A1, or the cell could migrate to different cortical layers and form different layers. Once a cell has reached cortical layers, there's another chance that the cell could die. That's the later stage apoptosis, apoptosis 2 in this model here. So one big question for brain development is, why are so many cells dying? We basically have around half of the neurons are dying during brain development. And this really sounds like a big waste of resources if you spend so much energy generating the cells and moving the cells to their correct position, to their correct cortical layer. So why do we have this large amount of cell death in the system? So we are running computer simulations with those different mechanisms, those different transition factors. We basically simulate how cortical layers are formed, starting with layer 6, layer 5, and so on. So over time, we basically have different cortical layers. We can look at the thickness of cortical layers. We can look at the cell density, how many neurons are in those layers. And based on this computer model, we can reproduce what we see in experimental studies. So we can reproduce the neuron density, neuron counts in human temporal cortex, wet hind limb area, mouse barrel cortex, macaque visual cortex, and so on. So we can tune our parameters to exactly reproduce what we see in anatomical studies. However, we can only do that if we include apoptosis or cell death. So if we don't have apoptosis, we are unable to reproduce what we see in actual brains. So cell death is really important if we want to have the specific organization that we see. So this can explain why you have this kind of large loss of resources, half of the neurons dying, is basically necessary to get the correct organization of cortical tissue. Our models can also reproduce brain disorders such as autism spectrum disorders, polymicrogyria, and subcortical band heterotopia in terms of the layer architecture we see for those conditions. And this model could have potential use for understanding what are the mechanisms leading to developmental diseases and what are potential drug targets if you want to avoid this kind of developmental pathway. We also looked at the role of the timing when you establish connections in the system. You can think about two extreme conditions. The first extreme condition is serial growth of the network, where you have one neuron extending an axon to other neurons and then stopping. And then the second neuron is starting, extending axons and stopping, and then the third one, and so on. So everything happens in a serial fashion, one neuron after the other, one axon after the other. The other extreme is a parallel growth, where the growth window is basically overlapping. So all neurons are extending axons at exactly the same time. And of course, those are extremes. In reality, you have something in between. You have sequential steps, and you also have things happening in parallel. But we're just looking at those extreme border conditions here. And what we find is that what we see in the computer model agrees with what we find in C. elegans, because in C. elegans, we also see that some neurons are born at the same time. They're able to establish connections at the same time. Other neurons are born in a serial manner. So first one neuron, then the other neuron. So we see different types of connectivity, both in the model and in the actual animal data in C. elegans. So if we look at the different modes here, we can look at distance between neurons and connection probability. We see changes in terms of how many long distance connections are established based on whether we have serial growth or parallel growth. If we have serial growth, we have more long distance connectivity compared to parallel growth. If we look at the percentage of bidirectional connections, so two neurons are connected in both directions, we have a higher probability that this happens if we have parallel growth in the system. And again, early starting neurons tend to become hubs. They have higher local efficiency, they have more long distance connections. And for parallel growth, we have more bidirectional connections and fewer long distance connections. And those are patterns we see in C. elegans. So this was all about how are connectomes changing. And the second part is really about how can we change connectomes, especially how can we change brain connectivity through brain stimulation. So if through brain development we end up with a connectome that has cognitive deficits or shows some symptoms, so if you have a connectome that leads up to schizophrenia, for example, is there any way to change the connectome to get it into a state that is more beneficial for patients? So we did a questionnaire with around 800 participants, half of them healthy and half of them with a brain or mental health condition. And we were asking them what kind of intervention they would prefer to change brain activity, brain connectivity, brain function. For the patients, many of them had Parkinson's disease, but we also had a range of other conditions, depression, post-traumatic stress disorder, schizophrenia, and so on. And we gave them an option of 5 different interventions. We had ultrasound, non-invasive brain stimulation, magnetic non-invasive brain stimulation, pharmaceutical drugs, electric non-invasive brain stimulation, and implants. And if we just ask them about implants, we see that the preferred intervention is ultrasound. Possibly people are just thinking about pregnancy scans. So if it's fine for pregnancy scans, then it's fine for brain and mental health conditions. Second place, we have magnetic brain stimulation. Third place, pharmaceutical drugs. So people know about drugs, but they also know about side effects. Then we have electric brain stimulation. Possibly people are thinking about electroshocks or the movie One Flew Over the Cuckoo's Nest, so it's not very popular. And last place is brain implants, Neuralink from Elon Musk or other devices. They're really, really unpopular with healthy participants and with patients. We see the same pattern. So in particular, there are many participants who say, I would never use it. So both patients and healthy participants. So why is this the case? If we ask participants about the perceived safety of different interventions, ultrasound is seen as the safest one, but implants are seen as the most unsafe option for brain stimulation or intervention in general. If we look at effectiveness of different interventions, ultrasound is seen as effective, but also brain implants are seen as very effective because you can directly stick an electrode into the brain, into a certain target region, so you can directly, locally target one part of the network. So even though both implants and ultrasound are seen as very effective, because of the different safety profile, the preference of participants is for ultrasound. So there was a huge need for developing this non-invasive ultrasound technology. How does ultrasound actually work? Ultrasound is a mechanical wave, like a normal sound wave, that is emitted from the transducer, some kind of speaker that's emitting the ultrasound wave. And the transducer has a curved surface. So the wave is coming out of the transducer and is then focusing in one particular spot, the focal area. And only in the focal area do you have enough pressure to have changes in brain activity. So what is happening in this focal spot? We have this incoming ultrasound wave. So we have changes in the mechanical properties of the membrane, which is then leading to changes inside the cell. So for example, mechanical deformation of actins. We have activation of mechanosensitive channels, changes in calcium influx, and then second messenger cascade. So we have changes in signal amplification, and then finally changes in voltage-gated ion channels in the membrane, and because of that, changes in terms of leading to action potentials later on. And depending on the parameters of the ultrasound wave, we can either increase or decrease the likelihood that neurons become activated. So we can increase or decrease the activity in our target area. Focused ultrasound is a relatively novel technology, relatively because the first paper appeared in 1953. Um, but the first device for human ultrasound came on the market in 2019. So there was a long period of hibernation where people were not using ultrasound. So it really just started again in the last 10 years that a lot of papers came up about ultrasound and a lot of people were joining the field. 2019 was also the time when the first international conference on brain stimulation with ultrasound was taking place. The nice thing about those devices is that we can directly stimulate deep brain structures without stimulating anything that is in the pathway. So we can stimulate hippocampus without stimulating any part of cortex that is in the way. There are several centers in the UK that have ultrasound stimulation devices, like on the left-hand side. Um, so Nottingham, Oxford, Cambridge, Plymouth, UCL, Glasgow, and there are more and more centers coming up. I think Bristol is about to get a system and so on. Um, at the moment, we have three systems in Nottingham, one in psychology, one in medicine, and one in the hospital. So the benefit is that we can be very focal in terms of where we stimulate. For the devices that people are using in the field, it's usually 1 to 2 centimeter size of the focus. If you have more expensive devices, you can go to a focal area of maybe just a few cube millimeters. So you can be even more defined than with the devices that we are using. The ones that we are using cost around £100,000. The ones that can go to a few cube millimeters cost around £1 million, and you can only use them inside an MRI scanner, and you have to shave the hair off. So there were some disadvantages as well. In general, you can reach deep brain structures directly: amygdala, thalamus, hippocampus, basal ganglia, cerebellum. So of course, this is very interesting if you want to look at Parkinson or dementia, for example. It's non-invasive, so we don't need any implants. You can increase or decrease brain activity depending on the protocol you're using. The device could be used for humans, for non-human primates, for rodents, depending on the device you're using. And importantly, we can measure the effect of brain stimulation while we're stimulating. Okay, so you don't have this interference of a mechanical wave with the measurement that you're getting. So we can measure fMRI while we're stimulating, or we can measure EEG while we're stimulating with ultrasound. So we had a review earlier this year where we want to understand how parameters influence the effect in terms of brain stimulation. So the theory is that if you have a higher intensity of your ultrasound and you have a lower duration and you have a higher duty cycle, you would basically expect that you get an increase in brain activity. On the other hand, if you have a lower intensity and a lower duty cycle, you would expect that you get a decrease in brain activity. So we reviewed 32 studies in humans, both clinical studies and studies in healthy participants, and both online studies where you measure effects while you're stimulating, and offline studies where you stimulate first and then you observe effects later on. And we also have an online database for this. So the idea is that this database can be updated in the future. So you get an overview of what studies are out there and what parameters were used for this study. So that's basically the setup for the review. And what we find is that results are a bit different to what would be expected. The blue curve here shows you what would be expected based on the biophysics. You have something called neuronal intramembrane cavitation excitation model. And based on this, you would expect that everything on the left side below the blue curve is reduced activity and everything on the right side is increased or enhanced activity. But you can see from the different studies and the different colors that that's not the case. And one potential reason for this could be that the response of a brain region also depends on what other brain regions are connected to it. So there might be some feedback loops that are changing the result at your target area. And there might also be local changes. So there might be a difference in terms of how many inhibitory cells and excitatory cells are there in an area. Or there might be different types of neurons, let's say phonoeconomo neurons, which you don't see everywhere in the brain, just in some parts of the brain. So there might be some local differences that can explain why the effect is not as expected. So what are the indirect effects of brain stimulation across the network? What happens if you stimulate in one region? How does it change activity patterns in other parts of the brain? Because what we want to do for stimulating the brain is often targeting circuits. Okay, for memory, you have the purpose circuit, for example, you have multiple brain regions that are involved. So what you would like to see is an effect that is not just in a single area that you're stimulating, but is also affecting other parts of the brain in a beneficial way for cognitive function. So to start with that, we can have computer models of what is going on in the system. So in this case, we are simulating global brain dynamics with Kuramoto oscillators, which is the simplest way to do it. So we have different brain regions with different oscillators. We are setting the frequency to be the same. So all regions have a 40 hertz oscillation, or all have 20 hertz, or all have 10 hertz. The coupling between brain regions is based on the weight of the fiber tract. So how many streamlines do you have between connected regions? And we also have delays in the system that are based on the length of fiber tracts. Mathematicians really don't like delays. They make models more complicated. But we do find that they make a difference. So it's more realistic to include delays in your models as well. And we can then measure changes in the system in terms of the global synchrony in the network. So we can calculate the mean synchrony, the average synchrony in the computer simulation. And we can calculate the metastability, which is the standard variation across time. And we can then try to use this computer model as a starting point to predict cognitive features in the brain. Because what we want to do on the long term is to stimulate the brain and improve cognition in patients. So we are wondering to what extent we can actually predict how network changes can improve cognition. So to link network changes with cognition, we first look at the correlation between network measures and cognition in healthy participants, and we start with topological measures. So what you see here is data from the Human Connectome project. We have 280 participants. We have different cognitive tasks at the bottom that are measured in all those participants. And we have different network measures. We have the clustering coefficient, C, for unweighted networks and for weighted networks, and the characteristic path length for unweighted networks and for weighted networks, and the edge density. And this is all based on structural connectivities, all based on diffusion tensor imaging, looking at brain connectivity here. And if we look at the correlation between those structural connectivity features and full IQ and crystallized intelligence, and then different types of fluid intelligence, processing speed, visual spatial ability, and memory, we see that correlations are not very good. Okay, so you see very few network features that correlate with cognitive features. On the other hand, we can observe spatial features of the network. So spatial features depend on the length of fiber tracks in the brain. They depend on the total volume of the brain. And those features give a much stronger correlation with intelligence measures, with full IQ and with different kinds of subscores. So we're looking at the convex whole volume, which is basically the volume of the brain. So you can wrap a foil around the brain, and you can look at the volume inside the foil here. So that's the convex hull volume. We can look at the distances between regions. So let's say if you have a region A that is connected to B and C, we can look at all those cases and we can then check, well, how far are B and C apart? Okay. So are B and C spatially nearby, which means they may be in the same module of the network, they may be both in the visual cortex, or are B and C far away in different parts of the brain? If B and C are nearby, you would say that A and B is integrating kind of similar information. Maybe A is integrating visual information. But if B and C are far away, then A might integrate different kinds of information. Okay. We can also look at this distance pattern if we only look at connections A, B, A, C that are long, so that are longer than the average length of a connection, or we can look at the distance between B and C where both connections are shorter than the average connection. And finally, we can look at the metric path length. So we look at the shortest path in the network, but the shortest path is based on how long are fiber tracks in length in millimeters. Okay, so we try to reduce the total length that you travel across fiber tracks for the metric path length. So for all those spatial features, we see much better correlations with IQ and with subscores of cognition. And finally, we can look at dynamic features of the brain. So we can look at the full IQ and all the subscores, and we can look at the Kuramoto model seeded with different frequency bands. So 40 Hertz oscillation like gamma band, 20 Hertz for beta, and 10 Hertz for alpha. And again, we see a very strong correlation, actually even better than for spatial features, in terms of how the metastability, how the variation is positively linked with cognitive scores. The idea for that is that if you have higher variation in the synchrony, if you have higher variability in the system, it's easier to switch from one brain state to another. Okay, so if you have a lot of fluctuation in your system, you can more easily go from one state to another compared to a network where you don't have much fluctuation. Okay, so the more fluctuation you have, the easier it is to switch tasks and to do more complex processing. And we basically see this kind of opposite pattern in dementia patients. So if we look at Lewy body dementia and we look at EEG and we look at brain states changing over time, we see for those patients that they are stuck in brain states for longer, which might explain why the cognitive performance goes down. Okay, so basically the fluctuation is a lot smaller. It's a lot more difficult for those patients to go to different brain states. And because of that, you have a lower cognitive performance in those patients. So this is a starting point. In the future, the idea is that those novel features like spatial features and dynamic features could be biomarkers to assess changes in brain disorders, but also to assess changes after interventions. So whether you have pharmaceutical interventions with drugs, or whether you have brain stimulation interventions or other kinds of interventions, CBT, and so on. So you can basically assess what changes you see and whether those changes might be beneficial for patients. And we currently have one PhD student in the lab who's basically looking at schizophrenia as a test case. So we want to understand whether we can predict both a cognitive impairment in schizophrenia, but also whether some of those features can predict positive and negative symptoms in schizophrenia patients. So that's ongoing work at the moment. So looking at brain stimulation, we started to look at ultrasound brain stimulation in healthy participants. So this is a study we published last year where we have a baseline scan. We have resting state fMRI, so we reconstruct functional connectivity, and we have a cognitive task, the stop signal task, that's very demanding. We have a green circle that is presented on the screen, and then you have to push a button once the green circle appears. But if the red cross is appearing, you have to stop yourself from pressing the button. And that's really challenging. So your fingers are already on the move, and then you have to stop it. So you get it wrong around half of the time. You're still pushing the button. We then plan the ultrasound experiment. So we plan where to put the transducer. We have another session where we stimulate a brain region. We look at changes in functional connectivity afterwards, and we perform the stop signal task again. And this task is performed around 1.5 hours after ultrasound brain stimulation. So we measure the reaction time, and we also measure the accuracy, how good you are in stopping yourself from pushing the button. We have two different targets. We have the thalamus and we have the inferior frontal cortex. And for the stimulation protocol, we use 500 kilohertz ultrasound, pulse repetition interval 200 milliseconds, duration 20 milliseconds. So we have a duty cycle of 10%, and the total duration of our stimulation is 80 seconds. Because of 10% duty cycle, this means we only stimulate for 8 seconds. Okay. So in total, we have 8 seconds of ultrasound stimulation. So what do we see afterwards, after stimulation? We basically roll participants over into the MRI scanner, do the structural MRI scan, and then we can start measuring fMRI and start to measure changes in functional connectivity. And we basically see changes in functional connectivity for other areas that are functionally linked to the stimulated area. On the left, you see the inferior frontal cortex, and you see other areas showing reduced functional connectivity after stimulation. And this reduced functional connectivity occurs earlier for the lingual cortex than for other parts of the brain. And it's interesting to see that this occurs many minutes after stimulation. So there might be some slow feedback loops going on in the system that only lead to changes later on. On the right-hand side, you see changes after stimulating the thalamus. So we see changes in supplementary motor area in different parts of the brain. And interestingly, we not only see reduced functional connectivity, but we also see for some regions increased functional connectivity, including for the contralateral thalamus in the opposite hemisphere. So we see this clear biological effect in terms of changes in functional connectivity. So the next question is, how is this linked to cognitive changes we see in the reaction time task, in the stop signal task? So we can look at the reaction time in the stop signal task. In yellow, you see the change when we stimulate the thalamus, and actually you don't see a change. So you have the same reaction time before and after stimulation. But if we stimulate the inferior frontal cortex in blue on the left, you see that reaction time goes down after stimulation. Participants basically get 8% faster in terms of their reaction time. And this is really good because if you think about other types of increasing reaction time. Normally, you get increases of 3 to 5%. So basically, being 8% faster is a big change in terms of the reaction time. And at the same time, accuracy stays the same. So it's not that people just push the button faster. They have the same accuracy as before. So it's basically observed for inferior frontal cortex, but not for thalamus. And we can predict the change in reaction time based on biological changes. So we can predict the reduction in reaction time based on the reduction in functional connectivity between inferior frontal cortex and postcentral gyrus. So the decrease of response time is significantly correlated with decreased functional connectivity. We can also predict the cognitive change based on the peak pressure we get into the target. So the more ultrasound pressure we get into the target, the stronger is the reduction in terms of reaction time. So both based on the biological effect and based on the physical effect, we can get a prediction of the cognitive effect. So this is a starting point to use network analysis to predict what will happen after brain stimulation. What effect could we predict to see? So one challenge with those methods is that the equipment is a bit expensive. It's £100,000 for the ultrasound stimulator, £50,000 for the new navigation device that makes sure that you're at the right position on the head. So £150,000 in total. So it's something that you can't really take home. You can't use it at home. But what we're also interested in is devices that can be used at home. For example, devices that are stimulating the vagus nerve. And this is a device here that is using ultrasound to stimulate the vagus nerve. It's the only company that is using ultrasound. There are lots of companies that use electrical stimulation. As a disclaimer, I'm scientific advisory board member of this company here that developed the device. So you can see on the left-hand side in green how the vagus nerve is innervating the ear. So this is the auricular branch of the vagus nerve. And this branch is coupled to the parasympathetic system of the vagus nerve. So normally, if you have electrical stimulation, you would target those patches you see in the middle in red. Okay, so that's where you would stimulate electrically, and those are the patches where we also stimulate with ultrasound. On the right-hand side, you can see the ultrasound stimulation device. It's like a headphone. You can put it on, and you can stimulate. We can stimulate for up to 30 minutes, but normally you could just stimulate for 5 minutes per day. So the device is already on the market as a wellness device at the moment. The idea is to get enough capital, venture capital, to basically run clinical trials and then to get FDA approval. But what do we see so far? There is anecdotal evidence that people sleep better and feel more calm and relaxed. But we have this pilot study here where we look at patients suffering from anxiety, 30 participants with a higher anxiety score as measured with the Beck Anxiety Inventory. They take the device home, they use it 5 minutes per day, and we then observe over 4 weeks how anxiety scores and other scores are changing. In general, we find that 85% of participants show significant changes in those scores. For the 15% that are not responding, it's not clear why they're not responding. It could be that they put the device on like a headphone. So if you have ultrasound gel on the device and you put it on the headphone, you might touch the hair, so you have all the gel in the hair, but nothing in the ear. The other possibility is that the shape of the ear is different for those participants and you don't have a good contact. This basically is not an issue anymore because for the second version of the device, we have better coupling material that can adjust to the shape of the ear. So that should be easier for the next generation of the device. So there might be a higher percentage of people that are responding. If you look at electrical vagus nerve stimulation, responder rates are much lower, so they're usually 40 to 60% responding. So what do we see for stimulation? We have the baseline score in blue before stimulation, and then the different scores after each week. And you can see that for the anxiety score, we have this significant reduction of 30 to 50% within the first couple of weeks. So the anxiety score in clinical terms goes from severe anxiety to mild anxiety. For some participants, it goes down to zero. And this curve is including the 15% participants where it doesn't work. Okay. So it's including all participants in the study. We also see improvements for post-traumatic stress disorder, depression, and sleep. So those are not patients that are classified as PTSD patients, but basically have some overlap in terms of the symptoms that you measure for those different conditions. For depression, of course, you have comorbidities. So you have many participants with anxiety who also have depression. So that could explain those results. You don't see big changes for sleep patterns. So that's the first study showing that there were improvements for anxiety. And we're currently running a randomly controlled trial in order to look into more detail in this. In order to do a randomly controlled trial, we need some kind of sham condition, some kind of control group. So for that, we developed a device that is a sham device. So you have an auditory sound indicating that the battery is on, same as for the active device. And you also have a heating up of the device over time. So if we have it on for 5 minutes, you wouldn't have heat up necessarily, but you would have heating up later on. So basically, we tested this in healthy participants, and healthy participants are unable to distinguish between active and sham stimulation. So we have almost complete blinding. So it's an ideal device for sham stimulation and for control group studies. So this is some pilot data where we have the sham device as well, and where we also have a comparison with electrical stimulation. So that was submitted for publication yesterday. This is looking at the emotional bias task, which is a task that can give you some indication of the mood of participants. This study is all in healthy participants. On the y-axis, you see the bias score. Higher score means participants are happier. You see the score before stimulation, during stimulation, and doing the test after stimulation, directly afterwards. We have active groups in darker colors, and in lighter colors, we have the sham stimulation. And we have electrical stimulation in blue and ultrasound stimulation in green. And what we see for vagus nerve stimulation is that for both electrical and ultrasound, we see improved mood during stimulation. For ultrasound, much stronger. And we also see improved mood after stimulation. But we don't see that to the same extent in the sham condition. So ultrasound is very effective for improving mood and actually more effective than electrical stimulation. But at the same time, for ultrasound, we have fewer side effects. So if we ask participants what side effects they observe, you have skin irritation and pain if you have electrical stimulation, but you don't have that if you have ultrasound. Um, just briefly, another challenge with brain stimulation is that people might like it too much and they might get addicted. So there was this term coined by Rob Henderson. I think he was in Cambridge for some time. So he termed this idea luxury belief, which is a belief that is beneficial to people who hold the belief, beneficial to the upper class and middle class, but has a negative cost for the lower classes. In a similar way, brain stimulation might be beneficial for the middle and upper classes, so it might improve performance. But in the lower classes, it might be used for addiction. Okay, so basically, this is like drugs, making you happier, getting your dopamine kick, or making you feel more happy. So there could be a cost in terms of new technology development. So we have to be sure to prevent the misuse of our devices, and we have some idea to implement some security features, making sure that you can't overuse devices that are being developed. So in order to develop devices, we are thinking about a closed loop stimulation, so closed loop ultrasound, but also closed loop magnetic stimulation. to make stimulation even more effective. So this is a 5-year MRC program grant that I'm leading, where we stimulate relative to the ongoing brain state in participants. So we basically have some idea what phase, what state the brain is on, and then we can stimulate with ultrasound or with magnetic stimulation at exactly the right time. So in the first 2 years, we will look at healthy participants, optimized protocols, and also use MR spectroscopy to observe the changes we see with ultrasound stimulation. In the following 3 years, we will look at improving cognition for depression and schizophrenia patients and improve motor circuits for Tourette syndrome. You can see collaborators here as well. So in summary, ultrasound neuromodulation is preferred by patients and the public. Transcranial ultrasound neuromodulation affects predominantly in a time-constrained manner spatially distributed regions that are functionally linked to the stimulated area. Biological effects are predictors of cognitive effects that persist for at least an hour following stimulation. There are some protocols that can lead to changes that want to have more permanent changes in patients. For auricular vagus nerve stimulation with ultrasound, it's painless. It shows a high responder rate. There's no need to have MRI scans before it. And there might be many applications, but we also see improvements in memory. So there's another publication in preparation. We already see improvements in mood, but then also in different types of memory, semantic working, and associative memory. And we also started a company a few months ago, Deep Brain. And the idea is to develop technology that is non-invasive, that is closed loop, that can stimulate deep brain structures directly, and that can be used at home. So I'm Chief Scientific Officer of that company. There's more information about brain connectivity and brain development, uh, healthy development and neurodevelopmental diseases and neurodegenerative diseases, and also the effect of learning and brain stimulation on networks in the book here. So that's the team here, alumni of the lab, collaborators and funders. You can learn more about what's going on in the lab by following us on X or Blue Sky. So thanks for your attention.
Thank you for a really fascinating talk. I can see lots of clapping in the chat. I'm just going to stop the recording.